What we built at Anderson is to try and replicate what essentially is a little biotech company working in a not-for-profit superstructure. But all the components that you would recognize in the biotechnology industry, whether you'd have research and development and manufacturing, as well as your regulatory affairs team, your clinical trials team, all these groups of people have to come together. And this isn't funded by the NCI or the NIH, but they can't get their mind around this type of science. It's too, it doesn't fit the typical funding model. They want to see tight bench research, and this type of translational research falls into what's been called the valley of death, really, because it's very difficult for folks to, at least at the federal government, to feel comfortable funding this type of science. But again, philanthropy, thank goodness, has really stepped in and recognized the power of doing this type of organization. So to move the field, we have to think for what the next part of it is. And we all have ideas, but one of the ideas we have is an idea that we can try and make T cells, not for every particular patient from their own blood, but choose one donor. So we'll choose Barbara as the universal donor, and we'll take her T cells and we'll put into all of you. And to do that, we've invented some technology that I think has some benefit at this point, where we've essentially want to make sure that there's no graft versus host disease. There's none of that unwanted destruction of the patient's normal body, but we want to preserve the anti-tumor effect with the chimeric antigen receptor. So we invented these little molecular scissors with a company called Sangamo Biosciences to take out genes, and we obviously have the fish in our trick that we can put into genes. So we can put genes in and take genes out. So again, this type of biology now is really right on the verge of being able to do these things.